I separate them?
Yeah.
There's a variety of ways. So, for example, I can, by changing the pH of the solution, sometimes I can break the bond between the antibody and the antigen. Sometimes I need to use other more stringent ways of doing that. So there's a variety of chemical tricks.
It's a very strong interaction, but it's not covalent.
Yeah.
So you need strong, really high salts, or a little bit of detergent, or something strong, but
now, there are some antibodies that bind so tightly and so well that you cannot elute them. So you have to add SDS to denature them, and then just dissolve everything.
But
So
the antibody
it's
is
not ideal.
covalently stuck to the bead, so that
Yeah.
doesn't come off.
Yeah. All right, great. So now notice that in this particular example, the protein that was bound by the antibody was the red triangle, the little triangles, right? It was not made at all against the blue squares, all right? So it should only bind, it only binds to the triangles, it does not bind to the squares. So when you precipitate, you should only precipitate the triangles, the triangle protein. But you can see that the blue square is itself attached to, probably let's just say by non-covalent bonds also, right, to the triangle protein. So now you have brought along the square protein together with the triangle. All right. So when you see that phenomena of other proteins coming along for the ride, we say that that is a co-immunoprecipitation. So immunoprecipitation refers to the direct protein being bound by the antibody, and co-immunoprecipitation is the precipitation of accessory proteins that are bound to the primary antigen. So in this case, the blue square. Okay, this video didn't work. Great. So here's a representation of that again. So the large circles are agarose beads, and there's the antibodies, and the antibodies bind, in this case, to blue circles. We spin them down, we wash the contaminants, and then we can elute and release them in the elution and analyze them for whatever we want to do. All right? Any questions? I have to go. So we can do that and run them on the gel and see how much of the antigen I get down, how much of the co-immune precipitated proteins come down. I can quantify them. I can look at what happens. I can combine this with fractionation. I can do, for example, a fractionation of RCC1, which is both in the cytoplasm and in the nucleus. I could ask, for example, is RCC1 bound by different proteins in either compartment? Maybe RCC1 in the nucleus is bound to something, protein X, and is bound by a different protein by protein Y in the cytoplasm. And maybe that's why it's in both compartments. So how can I answer that? I can get in the nuclei, I can do an immunoprecipitation from the nuclei, I can do a separate immunoprecipitation from the cytoplasm, and ask if there are other components coming as co-immunoprecipitates in either case. All right? So really the type of experiment that you do, then it's just, has to be carefully designed for the question that you are asking. There's not one particular set of steps that you have to follow. You can combine them in many different ways. This is what you need to learn to do, and this is what we're going to be asking you to do in homework and exams, all right? To be able to just put things together to answer a question. In all of these cases, we're assuming that the protein is abundant, all right? But sometimes proteins are really very scarce. There's very few amounts of proteins of those in the cell. So sometimes we have to enhance our sensitivity method. Sometimes, also when we are looking at the components of the cell, we are just looking at all the proteins that are present at that moment. And say in the case of RCC1, RCC1 may be made at some point, and then maybe it needs to, let me just imagine, mature for a couple hours in the cytoplasm before it can go into the nucleus. Maybe that's why it's in the cytoplasm and in the nucleus, because the new protein is just being maturing in the cytoplasm, and when it's fully mature, now it goes into the nucleus. How can we ask that? So we would need to discern the young protein from the old protein. So a way to distinguish old and new is accomplished by the process by the analysis of pulse chase. All right. So I want to go a little bit into the logic of pulse chase analysis. Again, the goal of a pulse chase analysis is to allow you to distinguish what happens to a particular protein through time, from the time that it was made and time thereafter. An equivalent version of that. It's gonna play? No, gonna play. Oh well. I do wanna. So let's take a minute break. You can stretch out, chat with your neighbors while I set this up.
I was writing down the answer to the third question when she called you, and I was like, yeah, but I was like, I remember the first time I was like, he was like, yeah, I was like, I don't know. He was like, I did like, I'm not surprised. I cannot be the one. But I was like, I turned and then I said, and then I heard my name and I was like,
Okay, we're back. Thank you. So this is the idea of the pulse chase. So in a regular cell, you have messenger RNAs being translated by ribosomes all the time, right? So how much you make is going to be dependent on the abundance of that messenger RNA. All right? So the idea of a pulse is that we can feed the cell amino acids that are labeled with radioactivity. So in this case, with those red dots. So that means that when the cell picks up those radioactive amino acids, is going to incorporate into the proteins made during that time that you fed the cells the radioactive amino acid. So those proteins are going to exist in that radioactive state for as long as radioactivity lasts, right, which is a long time. And then we can add non-radioactive amino acids in later on, and that would constitute a chase. So we're washing away the radioactivity, the radioactive amino acids, free amino acids, replace them with non-radioactive amino acids, and then the proteins that are made after the chase are not going to be radioactive. So when you look in the cell, if you had magic glasses and look in the cell, the radioactivity would be present only in proteins that were synthesized exactly in that window of time that we call the pulse. The proteins that were made before that pulse are not radioactive. The proteins that are made after the pulse are not radioactive. And if we're looking for radioactivity, that means we are only seeing a very small subset of proteins, only those that were made at that pulse moment. And that's really powerful, because now you can forget about who came before, who came after. You can ask, what happens to this cohort of proteins? All right? It would be like if in this university, we wanted to follow what happens to the class of 2022. We are not going to be looking at the students that came before. We're not going to come to look at the students that came after. We're only going to be looking at that freshman class. And then we're going to track them to see as they go from their freshman to their sophomore, etc. years. All right? That's the question you're asking with a pulse chase analysis. All right? Clear? Now, to do this with a whole cell extract, so if you were just to say, okay, I'm going to take the whole cells right after the pulse, and then in our hourly intervals after the pulse, you're going to get a mess, because the cell at every moment is making thousands of different types of proteins. So all of those that are made constantly are going to be made at that pulse time, and they are going to be present thereafter. So it would be like, looking in the university at all students from all colleges, from all majors. That would be a mess. Maybe we are interested in the biology majors only. So we would not be analyzing the entire population. We want to select only for those majors. So how do we select for a specific subset of proteins? You know the answer. With antibodies, right? So we use our antibodies to grab that group of proteins, that group of students, and then pull them out from the rest. Right? So we would have to use an immunoprecipitation to select out of the mess of all radioactive proteins only the protein that we are interested in studying. Clear? So this has to be ultra clear, otherwise you will not understand at all what's coming next. So think about it for a second, and then ask questions if you have them. Yes?
Is there a time limit on this kind of analysis regarding radioactive decay?
A little louder.
And so is radioactive decay a problem associated with this analysis?
Not really. I'm going to blow one of my questions down the road, but the radioactivity that is used decays in a matter of weeks. So you're going to be the experiment within a couple of days, so there's not going to be a decay problem.